Dear students, today let us learn about single cell and cell suspension culture. Let us start with the introduction to single cell culture. Single cell culture is the method of culturing isolated single cell of plants aseptically on culture medium under controlled conditions. The culture of single cell provides an ideal system for studying cellular metabolism and other processes, or to form a clone of cells, or to regenerate whole plants. Let us now see isolation of single cells. Single cells can be isolated either from suspension cultures or from plant organs. For single cells isolation from suspension cultures, the cells are separated by suitably filtering out the cell clumps and harvesting the cells from the suspension of cells by centrifugation. Single cells can also be isolated from plant organs like leaves by mechanical or enzymatic means. In the mechanical method of isolation, small pieces of leaves, approximately about 1 centimeter square, are ground in a suitable culture medium using pestle and mortar, and the cells are pelleted out by centrifugation of the resulting homogenate after filtration. While in the enzymatic process, the lower epidermis of leaves is peeled off and moderate pieces about 4 cm square of leaves are incubated in a mesogyme, which contains about 0.5% of the enzyme, or pectinous solution containing a suitable osmoticum like 0.3 molar mannitol, since the enzyme treatment tends to weaken cell walls. Next, let us take up the culture of single cells. Isolated single cells normally fail to divide in the basic tissue culture media, and a nurse tissue or a conditioned medium is generally used for the culture of single cells. A conditioned medium is a medium obtained by filtering the medium in which plant cells have been grown at high cell density for about 24 to 48 hours, and it contains nutrients secreted by the cultured cells. On the other hand, a nurse tissue is a callus tissue on which a single cell is cultured. Single cells can be cultured using the following techniques. The first technique for culturing the single cells is filter paper raft nurse tissue technique. In this method, the single cell is cultured on the surface of a raft made of filter paper, which is placed above the callus tissue called the nurse tissue. The single cell absorbs nutrients from the callus through the filter paper and divide to form a mass of cells known as a single cell clone. The second technique for culturing of single cells is the microchamber technique. This technique involves the culturing of a drop of liquid medium containing the single cell on a sterile microscopic slide and creating a chamber by placing 2 cover glasses on either side of the drop and another cover glass on top of the 2 cover glasses. The third method is the microdroplet technique. In this technique, Single cells are cultured with a drop of liquid medium in the walls of inner chamber of a special cuprac disk surrounded by outer chamber which is filled with sterile distilled water to maintain the humidity. The next technique is Bergman's plating technique. The technique was developed by Bergman in 1960 and is the most popular method for growing single cells. The technique involves mixing a suspension of single cells with an equal amount of melted agar medium cooled down to 35 degrees Celsius on a petri dish. The medium containing the single cell suspension solidifies on cooling and the petri dish is kept at inverted position. The next technique is the thin layer liquid medium technique. Single cells can also be cultured in a thin layer of liquid medium. It is not possible to follow the development of individual cells in this method since the cells are not fixed in position. Let us now see the importance of single cell culture. The first importance of single cell culture is single cell cultures can be used to obtain single cell clones and regenerate plants from the clones. The second importance is since high degree of variations is present in cultured tissues, the genetic variability can be exploited for crop improvement programs. Next, the isolated single cells can be used to regenerate mutant genotypes since they can be handled easily as microbial system for induction of mutation and cell selection. The next importance is single cell culture can also be used for selection of cell lines that produce higher amounts of secondary metabolites compared to the intact plant. The next importance is cultures obtained from single cells have also been used for biotransformation or the cellular conversion of an exogenously supplied substrate compounds to another compound in higher amount. Let us now take up cell suspension culture. A cell suspension culture is the culture of single cells, small cell groups or larger cell aggregates in a liquid medium under agitation and aeration. Cell suspension cultures are generally initiated by culturing small masses of callus tissues in liquid medium, which is then agitated during the culture period. Let us now study about the growth curve of plant cell suspension culture. The growth curve of a typical plant cell suspension culture consists of four essential stages: an initial lag phase, an exponential or logarithmic phase, linear phase, and stationary or dead phase. The lag phase is the initial period of slow growth where the cells adjust themselves to the nutrient medium and is followed by logarithmic phase characterized by rapid cell division causing a logarithmic increase in cell number. The cells then enter the linear phase of growth where rapid cell division results in a linear increase in cell number. Finally, the rate of cell division within the culture declines and it enters the stationary phase as nutrients are depleted in the medium and some cells of the culture becomes senescent, causing death. The duration of each phase of growth is dependent on the species or genotype selected, explant use, culture medium, size of the inoculum, and the time of subculture. Let us now take up measurement of cell growth in suspension culture. Growth in such cultures can be monitored by the following methods. The first method is determination of cell number. The increase in cell number in cultures can be counted with a hemocytometer under a microscope. The second method is determination of packed cell volume. The packed cell volume at different stages of culture can be measured by harvesting definite volumes of cell suspension and measuring the volume of cell pellet after centrifugation at 2000 xg for 5 minutes. The third method is determination of cell fresh weight and dry weight. Growth can also be measured by determining the fresh weight of harvested cells or as dry weight of cell mass by drying the pellet in a hot air oven at 60 degrees Celsius for about 12 hours and weighing the dried cell mass. The next method is by determination of optical density or OD. Growth can be measured by measuring optical density or OD of the culture at different time intervals as the cells increase in number during growth, causing change in turbidity of the medium. Let us now learn about the test for viability of cells. Cell viability can be observed by observing protoplasmic streaming and the presence of an intact nucleus under a microscope. Cell viability is also commonly analyzed by using stains like 2,3,5-triphenyl tetrazolium chloride or TTC, fluorescein diacetate or FDA, Evans blue, etc. Fluorescein diacetate or FDA is the most frequently used stain, and when cells are stained with 0.01% FDA, the dead cells fluoresces red when observed under a fluorescent microscope. Evans blue also stains the dead cells blue as soon as the cell suspension is mixed with 0.01% of the stain, while the viable cells remain unstained. On the other hand, 2,3,5-triphenyl tetrazolium chloride or TTC stains the living cells red due to enzymatic reduction of the white stain to red, 1,3,5-triphenyl fromagen, while the dead cells remain white. Another test for cell viability is to determine the mitotic index, which can be calculated as the number of nuclei in mitosis out of the total number of nuclei observed multiplied by 100. Rapidly growing cell suspensions generally show mitotic index of 3 to 5% on an average. Now let us take up types of suspension cultures. There are two types of suspension cultures: batch culture and continuous culture. First, let us learn about batch culture. Batch culture is a type of suspension culture where the cells are cultured in a small finite volume of agitated liquid medium in culture vessels. The cultures are subcultured at the stationary phase by pipetting out a small volume of the suspension and transferring to fresh medium. Batch cultures are again of the following types: The first type is the slowly rotating culture. In slowly rotating culture, cells are grown in a specially designed flask with nipple-like projections. When these flasks are loaded in a circular manner on a large flat disc of a vertical sucker maintained at 1 to 2 rpm, the cells within the nipple are alternatively exposed to air and bathed in the culture medium. The second type is the shake culture. It is the most common and effective method of suspension culture where the cells are grown in conical flasks mounted on the platforms of orbital suckers maintained at a speed of 60 to 180 rpm. The third type of culture is the spinning culture. In this method, cells are cultured in large volumes in 10-liter bottles which are rotated in a culture spinner at 120 rpm at an angle of 45 degrees. The fourth type of batch culture is the stirred culture. This is also a method for large-scale batch culture where the cells are grown in large containers which are not second but the contents are stirred continuously by bubbling sterile air or with the help of an internal magnetic stirrer that revolves at 200 to 600 rpm. The second type of suspension culture is the continuous culture. A continuous culture is similar to stirred culture, but the cell population of the continuous cultures are maintained in a steady state by regularly replenishing the used or spent medium with fresh medium. Continuous cultures can be either closed or open type. In closed continuous culture, only the spent medium is replaced by fresh medium, while in open continuous cultures, some amount of both cells and the used medium are replaced by equal volume of fresh medium. The open cultures are further of two types, chemostats and turbidostats. First, let us see what chemostats are. In a chemostat, a nutrient is added to the medium which becomes growth limiting while other nutrients are still in concentration higher than required and the effect is reflected in cell growth. Such systems are ideal for the determination of effects of nutrients on cell growth and metabolism. The second type is the turbidostats. In the case of turbidostats, the cells are allowed to grow up to a pre-selected turbidity measured as optical density or OD or pH by a turbidostat before replacing with a predetermined volume of the culture by fresh medium. Next part is the synchronization of suspension culture. A synchronous cell culture is the culture in which most of the cells proceed through the cell cycle phases like G, S, G2 and M simultaneously. However, cells in suspension cultures are mostly asynchronous as the cells in suspension cultures vary greatly in shape, size, cell cycle time and nuclear DNA content, and this property limits the use of cell cultures for studies of biochemical, genetic, physiological and other aspects of cell metabolism. Different methods are used for synchronization of cell suspension culture. Physical methods. Under the physical methods, let us first study about the first method, that is selection by volume. In this method, synchronization is achieved by selecting the cells on the basis of size of cell aggregates using cell fractionation. The next type is temperature shock. This method uses low temperature shocks combined with nutrient starvation to induce synchronization of suspension cultures. The next group of methods belongs to chemical methods. The first type of chemical methods is the starvation method. In this method, synchronization of cells is induced by starvation of an essential growth compound like growth hormone. The next type of method is inhibition. Synchronization by this method is brought about by using a biochemical inhibitor like inhibitors of DNA synthesis, for example 5-aminouracil, 5-fluorodeoxypurine, hydroxyurea or excess thymidine that temporarily block the progression of events in the cell cycle and accumulate the cells in a specific stage. The third type of method is the mitotic arrest method. Cell cultures are also synchronized by treating with 0.02% weight by volume colchicine for 4 to 8 hours, which inhibit spindle formation. Let us now take up the importance of cell suspension culture. Firstly, suspension cultures can be used to study significant events about cell physiology, biochemistry, and metabolism at the cellular level. The second importance is suspension cultures can be used for production of useful secondary metabolites. The third importance is cell suspension cultures can also be used to produce mutant cell clones by mutagenesis, from which mutant plants can be regenerated. Let us now see the conclusion. Plant single cell and suspension cultures are important systems for the analysis of complex physiological processes at the cellular and molecular levels and for producing single cell clones. Plant cell suspension cultures also provide a source for production of useful secondary metabolites, and mutant plants can also be regenerated from the cells.